Hello class, today we're going to be talking about cell reproduction, okay? So there's a couple goals we're going to have to go ahead and go over today. We're going to be able at the end of this lesson to distinguish between chromosomes, genes, and traits, describe the three stages of interphase, that's a typo, I apologize, the three stages of interphase, discuss the behavior of chromosomes during mitosis, that's the division of cytoplasm, prophase, metaphase, anaphase, telophase, and cytokinesis. Then we're going to describe what happens when there is uncontrolled cell growth that can lead to cancers and tumors, and then understand what binary fission in prokaryotes is. So, first question to even start this is ask yourself, why do cells need to reproduce? Well, for one reason, in multicellular organisms, we actually depend on cell division for development for a fertilized egg, for growth, and for repair. Think about it in the sense of, let's say you cut your skin and you have a paper cut. Well, those skin cells need to reproduce and actually create clones of themselves so that we can come and go ahead and heal that part that's actually injured. So we need to be able to actually clone our body cells, okay? So this is a picture right here of a zygote. This is after fertilization has occurred. So once the sperm and egg come together, it is considered a zygote. Well, from that zygote, you actually have to multiply and multiply and multiply, and this is where mitosis is seen. So from a zygote, fertilization has occurred, so half of the chromosomes come from mom, half of them come from your dad, and then your cells start replicating. So one cell becomes 2 cells, becomes 4 cells, becomes 8 cells, becomes 16 cells, and so forth and so forth. So you get the idea. Until eventually it's actually making a whole organism. For this case, this is actually a zygote of a sea urchin, from a zygote all the way until it's an actual full organism itself. So some terminology before we get started. What is a genome? A genome is all the genetic material of an organism. Somatic cells are non-sex cells versus gametes, which are sex cells. That X and the Y, those are your sex cells, okay? But somatic cells, non-sex cells, those are going to be all other body cells. So hair, skin, nails, you name it, organs, everything else in your body besides sex cells are known as somatic cells. Okay, somatic cells, or non-sex cells, they actually have 46 chromosomes in them. So right here, you see a karyotype, and you can see all 46. So it's 1 chromosome, 2, 3, 4, 5, 6, 7, 8, 9, 10, and so forth, all the way till you count all the way to 46 chromosome pairs. The only thing that's different, or how you could say this differently, is by saying instead of 46 chromosomes, you can see it is 23 pairs. So here's 1 pair, 2 pairs, and 3 pairs, and so forth. Gametes, on the other hand, okay, when a sperm meets an egg, obviously you don't want to have 46 and 46 chromosomes. So instead, gametes, or sex cells, have half the number of chromosomes. So they only have 23 total chromosomes. So that way, when a sperm and egg come together, guess what? They actually are going to make one full cell of 46. That is known as haploid, because diploid is 2 sets of chromosomes. Haploid means that there's only 1 set of chromosomes. So a little bit of background. There is a woman by the name of Henrietta Lacks. Okay, they actually call, and she's known for having immortal cells. What does that mean? It means that basically she was a woman that they actually performed a biopsy on her because she had cancerous cells. So they removed that cancerous cell tumor, or the area that had actual cancerous cells, and they were actually able to go ahead and put it into a tube and actually grow it with nutrients and whatnot. They were actually able to basically allow the cells to replicate and replicate and replicate, because cancerous cells, they actually form tumors because it's uncontrollable cell growth. So those cells are not basically controlled like normal cells are. They're basically growing and growing and growing. So that's why they actually form tumors. Well, this woman's actual cells were actually used and still are being used to this very day, because at the time when she actually had the biopsy performed back in 1951, she died later that year in 1951 as well. Her cells are still around and still being grown in laboratories today, because they're trying to go ahead and investigate cancer and trying to find viable ways to go ahead and hopefully prevent cancer from occurring. There's 2 types of cell division. There's mitosis, which we said happens in all somatic cells. It is when there is going to be a division of the nucleus, and At the very end, you actually have cytokinesis, which is complete division of that one cell into two cells because it completely divides the cytoplasm altogether. Second type of division is meiosis. This is the one that has to do with sex cells. So basically, you're going to have sex cells in your ovaries or your testes if you're a male. Sex cells are produced after a reduction in chromosome number. So let's go ahead and jump into mitosis first. This is what we're going to be talking about in this lecture. Is this first part is mitosis, okay? Next chapter is going to be meiosis and sexual reproduction. So this is actually the entire cell cycle. So the somatic cells or your body cells, they're going to undergo mitosis, okay? But before they go through mitosis, which is this yellow L right here, they have to go through interphase. Interphase is this right here, and right here. So this entire other part, like 3/4 of this entire cell cycle, is interphase, okay? Interphase, they have G1, which is growth phase, where basically the cell's going to grow and grow and grow. Then DNA replication occurs, okay? And last but not least, there's growth and preparation for mitosis that occurs here. So during interphase, okay, which is 1, 2, 3, like I said, there's G1, growth phase. There's DNA synthesis replication occurs here. Basically, it's replicating the DNA. And last but not least, G2 phase is more and more growth and preparing for division of mitosis. So interphase right here, G1, S, and G2, okay? Then if it passes all the checkpoints, everything's good to go, your body says, yes, this cell can divide, then it'll begin mitosis, which is this yellow arrow right here. This is the mitotic phase. You have prophase, metaphase, anaphase, and telophase, and last but not least, cytokinesis during mitosis. Okay, I like to memorize it as PMAT, P-M-A-T, and then you just have to know cytokinesis is at the end. So let's go ahead and start and talk about what happens in interphase. Remember, it's the S phase for DNA synthesis, which basically means if you have one single chromosome, at the end of synthesis or DNA, that S phase is going to become two sister chromatids, okay? So start off with one single strand, result in two sister chromatids. So right here is our single strand that we're going to start off with, and right here is a good cartoonish picture as well. Then after DNA synthesis occurs, you have two. It makes a complete identical copy of itself right here and right here. So this is considered one chromosome. This is also considered one chromosome. The reason they say it's one chromosome is because each of them have a centromere, which is in the middle part right here. The centromere on this one in particular is actually connecting the two sister chromatids. So once it replicates itself, we have one sister chromatid on this side and another sister chromatid on this side, okay? They're joined once again at that centromere portion. I'm going to go back up real quick. Also, one thing you want to notice, like I said, this is a centromere, the very center of the actual pairs of sister chromatids. That's where they're attached. Don't get it confused though with kinetochore. The kinetochore, as you guys can see right here, is these blue or this greenish area right here on this one right here on the right hand side. That greenish little thing that's on the side of the centromere, that's going to be, imagine it like handlebars, okay? That's the kinetochore. It's handlebars where the spindle fibers are going to attach to it to pull them apart when we get into that phase, the anaphase, okay? But this is the kinetochore right here, which is the green, like I said, imagine them like handlebars. But the center is still called a centromere. So homologous pairs of chromosomes have the same length, shape, and genes located on them, except for the X and Y. So what does that mean, homologous pairs? So I just use this one for example. This chromosome 5 came from mom, let's say. This chromosome 5 came from dad. They're going to have, as you guys can see, they're the same length, okay? They're going to have the same shape, just like how you see chromosome 4 right here has a little niche right here, right here. They have the same length, same shape, and they're going to have the same genes on them. So let's say right here in this pinkish area is a gene, let's say for hair color. It's going to be in the exact same loci right here on the other chromosome 5 from the mom or dad. So that's what homologous chromosomes mean, okay? One from mom, one from dad. Like we said though, they're all going to be homologous except for the X and Y sex chromosomes, okay? Reason being, in a female, we would see an X here and an X here. But in males, we see an X and a Y. As you guys can see, they're completely different chromosomes. The X carries a lot more gene information compared to the Y chromosome. Okay, the X is going to be coming from the actual mother. The Y is the one that would be coming from the dad. So that's why they say the dad is the one that determines whether you're going to have a girl or a boy. Because the only one that can give you a Y to become a boy is going to be your dad. So once again, single chromosomes right here. They're homologous because let's say this is chromosome 1 from mom, chromosome 2 from dad. After DNA replication occurs in the S phase of interphase, then you have pairs of homologous chromosomes. Because right here, this one replicated. Remember, this is chromosome 1 from mom, but it got replicated. And this is also chromosome 1 from dad, but it got replicated. So that's interphase. This is what it's going to look like during interphase right before it gets to mitosis, okay? Remember, this takes up about 3/4 of the entire cell cycle. So it's a lot of the time cells are in interphase. So this is actually what it actually looks like in a real cell. So as you guys can see right here, you can't really distinguish any kind of chromosomes yet. There's nothing really darkened in because at this time, the actual DNA is in the form of chromatin, which is just basically very, very loose DNA. It still has the histone proteins in them, but just imagine like loose spaghetti here. That's what's in interphase. The chromosomes are not tightly wound up yet until you get to the S phase, which is synthesis phase. But you still really can't distinguish anything that well. It's the G1, the growth phase, the S phase where DNA needs to replicate, and last but not least, G2 phase again. So I like to imagine this, if you're like a visual person, is let's say I wanted to make an identical copy of myself. What I'm going to do is I'm going to eat and eat and eat and grow and grow and grow, okay? After I grow and grow during my G1 phase, then I'm going to replicate my DNA inside of me because when I get split down the middle, I need half to go to my old cell or my old self and then half to go to my new self, my clone self, right? Then I'm going to need to grow a little bit more because I still need to be able to cut myself completely in half. So let's say I just grow and grow and grow and I got really, really big, like double my size. So let's say if I weigh 100 pounds right now, then what I'm going to do is I'm going to grow. DNA replication occurs. I'm going to grow some more until I get to 200 pounds. So I doubled my size. Then and only then, if everything checks out great with my checkpoints, I'm going to go through mitosis and then go through all the phases. In the very end of mitosis, I get completely split into half during cytokinesis. And now I'll have an identical clone of myself, my original self, an identical clone of myself. Now that we're done with interphase, which we have to go through before we begin mitosis, now we're ready to get into mitosis. So here's interphase once again. G1, S phase, and G2 phase right here. Okay. Like I said, the cell is actually going to go through checkpoints and double check to make sure things are done correctly. For example, to make sure, did your DNA replicate correctly or were there mutations? Because if there's mutations, we want to go ahead and kill that cell or stop it from dividing because we don't need a cell that's mutated to continue to divide because that's where cancer comes about, okay? Usually, if there's a mutation or something happened where it did not duplicate correctly during synthesis phase, this will actually shut it down right here in this checkpoint. Your body is like, nope, we do not want it to continue on. We're going to go ahead and cause apoptosis to occur, which is cell death. And if it doesn't get stopped at this checkpoint, then pretty much you have a mutating cell replicating. And unfortunately, it can usually lead to cancer, okay? Because there's actually nothing controlling it from replicating faster and faster. So let's go ahead and start with the different phases of mitosis. First phase is prophase, okay? Chromosomes condense, so right here, they actually get that loose spaghetti I told you that happens in interphase when it's chromatin. Now it's really, really, really tightly wound up DNA and that histone proteins. So imagine I gave you loose spaghetti in interphase, but now I let the spaghetti sit out for a while and I put it in a plastic tube and I dried it out. So now instead of all that loose, wet spaghetti, I'm giving you hard, dry spaghetti. Basically, the chromosomes have really, really condensed the DNA because now it's going to need to move around to get separated. So we need it nice and tightly compact. We don't want to lose any of those noodles. So prophase, as you guys can see here in the actual picture, you can start to see chromosomes. You can't see them clearly, but you can see darkened areas, which means the chromosomes are actually condensing. Another thing that's key here is you'll actually see the nuclear envelope starts to break down. And then you'll see spindle fibers start to form. These guys are right here, attached to the centromeres right here. And inside of a centromere, these little circles are centrioles. Okay, they're starting to move to opposite poles during prophase. So you guys can kind of see these spindle fibers starting to form right here in this area, and also in this area right here. I can see little spindle fibers, but all in all, I can also see this condensed chromosome right here, and it even tells you in this one right here, chromatin condenses into chromosomes. So now we can actually say there is actually chromosomes that are starting to appear. Then there's prometaphase, which is just right before metaphase. Right here, the kinetochores appear at the centromeres. Remember, the centromeres are in the middle part of the chromosomes, that middle area. And kinetochores are the handlebars, remember, that attach to sister chromatids. So here, kinetochores appear around the centromeres of the sister chromatids. Microtubules start to attach to those kinetochores, and everything's beginning to kind of guide their way and line up towards, the chromosomes are lining up towards the middle, and the actual spindle fibers and the centrosomes are actually going to opposite poles there. So right here, you can see the centromeres, one here. They were starting really close together. Now one's on this side, one's completely on the other side here. They're getting ready for metaphase. Metaphase is going to be where they line up on the metaphase plate. So the metaphase plate is the center imaginary line down the center of the cell. Homologous chromosomes or sister chromatids are lining up here on the metaphase plate, and microtubules are now completely attached to those handlebars, those kinetochores, on the sister chromatids. So here's the cartoon version. You can see the chromosomes all nicely lined up on the metaphase plate. Here's our spindle microtubules, spindle fibers, microtubules right here, attached to those kinetochores on the chromosomes. And here in this circle area is my centrosome. Remember, those little guys inside, they are our centrioles. Okay, and right here in this actual real life cell, which you'll see in lab, this is what it looks like. So as you guys can see all these little lines right here, these are actually the spindle fibers on this side as well. And this kind of darkened area, it kind of looks like a starfish, like a blast. That's where our centrosomes are, these guys right here. Then next is anaphase. Anaphase, the sister chromatids are getting pulled apart. Remember, the kinetochore have microtubules attached to them. Now they're going to pull them apart to actually get one chromosome, sister chromatid on each side of the poles. So right here, you can actually see them getting pulled. These are little chromosomes getting pulled that way, and these are getting pulled this way. This is the cartoon version. These are sister chromatids getting pulled this way to this pole. These are getting pulled this way. Really, really good picture of an actual cell, of a cell that you're going to be seeing in lab right here. Telophase. Telophase, now that they're getting pulled to the different sides, you're going to be able to see two daughter nuclei begin to form at opposite poles. The nuclear envelope right here starts to reform because remember it broke down in prophase. Now the nuclear envelope starts to reform here. Chromosomes start to also uncoil because remember they got really, really tightly wound up, those DNA and the histones. Now they're going to start loosening up little by little, and daughter cells start to look genetically identical at this point. And the last phase is going to be cytokinesis. It is actually when the cell, remember it was kind of forming these two little circles like this, and then now we're going to have this pinch off and pull apart completely and completely separate the cytoplasm of both cells. Okay, in animal cells, it forms a cleavage furrow. Right here, you can see the cleavage furrow right here and right here. Literally, it's called a cleavage furrow. So right here, it's going to be a boop, and it pops off, and it makes two new cells. For plant cells, though, obviously, they have a cell wall. So at this stage, they're not going to form a cleavage furrow. For the plant cell, they're going to start forming a cell plate to separate these two plant cells right here. And a little bit of recap on mitosis. First, we had interphase. Remember, interphase takes up about 3/4 of the actual cell cycle. It actually has 3 phases itself. Interphase consists of G1, which is the growth phase, S phase, which is DNA synthesis or DNA replication, and then we also have G2 phase, which is another growth phase before it gets into mitosis. Then we actually have prophase right here. Remember that chromosomes start to really, really tightly condense. We also have the nuclear envelope start to break down. And spindle fibers are actually they're actually attached to the centrosomes, and they're starting to pull away to opposite poles right there. Okay. Then the next phase, remember PMAT. So prophase, metaphase is next. Metaphase is when all the sister chromatids or chromosome pairs line up right here in the metaphase plate, and all of the microtubules are attached to the kinetic cores of those sister chromatids. Okay, everything's lined up on this metaphase plate right here, or the equatorial line. And this is a really good picture right here. I like that picture a lot. Then we have anaphase. Anaphase, sister chromatids are getting pulled apart to opposite poles. This is a really good representation of right here. And last, telophase. Telophase, remember, they're actually forming two daughter nuclei. There's the nuclear membrane that starts to actually reform around each of those sets of chromosomes on each cell. And last but not least, then you have cytokinesis. Remember, in animal cells, we have a cleavage form that's going to completely separate them and cut that cytoplasm down the middle. And in plant cells, it's going to actually start creating a cell plate. So for each of these, you're going to want to know what and be able to put, I would write this down in a table and use those study guides. And you're going to want to write down what happened in each of these phases. So in interphase, what happened in G1, in S phase, and in G2 phase, okay? And after interphase, we have mitosis. So what key items happen during prophase, prometaphase, anaphase, telophase, and cytokinesis? And can you tell the difference between cytokinesis in an animal cell and cytokinesis in a plant cell? This is the end of part 1 of this lecture.